Hello everybody, welcome back to our review videos, this time for Unit 2, all about the cell. So this video in particular is going to be focusing on what defines a cell. It's going to be focused on the cell membrane, what gets in and out of the cell, and is going to focus on how cells integrate together to form tissues. What it is not going to cover is going to be transport, osmosis, endosymbiotic theory, or organelles. Those are going to be separate videos, so we're chopping it up to be a little bit more digestible for this unit. Now, one thing about this unit is it is what I call a foundational unit. So the material we learned here is a lot of definitions, it's a lot of language that maybe we're not familiar with, that we're going to have to start being familiar with to be successful in all of our other units. So it's very vocab heavy, which is why I've made you guys Quizlets. And by vocab, I don't just mean like words and definitions, I'm also talking about certain proteins and certain structures that you just sort of have to memorize. That is not my favorite way to learn, but unfortunately it is what we're going to have to do in this unit. So please make sure that you are practicing with those Quizlets, or make your own if you prefer, and make sure that you're reviewing your notes so that we have all of these words down so that we're successful for the rest of the year. So to start off this review, I always like to give you some learning objectives. And remember, if you can do these, you should treat them as like a checkpoint. If you can do it, then you're done. You don't have to study that anymore. If you can't, then you need to go back and make sure that you can really nail that down before you keep moving on. So for this review, I need you to be able to understand how a cell is defined both by cell theory and the features that identify a cell. You need to be able to describe the structure and function of the cell membrane, including but not limited to the phospholipid bilayer and the different types of membrane proteins. You need to be able to explain which and why some molecules are able to pass freely, while others are going to require a protein and or energy to pass. And then you're going to have to be able to describe how cells form tissues. And we're going to talk about all that in the review, but maybe you already know some of this, check it off, and then you can sort of skip around in this video. So in a unit called the cell, you can imagine it's very important that we're able to define what a cell is. And one way that we define a cell is through cell theory. So this is 3 parts. Yes, you have to memorize all of them. They're all sort of related, so it's not too difficult. But the first part is all organisms are composed of cells. The second part is cells are the structural and functional unit of all living things. Those are sort of interchangeable, right? 1 and 2. And then 3 says cells come from pre-existing cells. So in class, I gave you the example that your skin cell, when it dies, or skin cells are always going to be replacing themselves with other skin cells. So it comes from other cells. And even you came from 2 cells. You came from mom's egg and dad's sperm originally. Now, that is sort of our cell theory definition. We also have our hallmarks of a cell, which are in your notes. But hopefully by now you're able to name all 4. 1, cell membrane, which we're going to talk about. 2, a nucleic acid. DNA is typically our genetic material, but all cells also have RNA in them as well. Then we have the cytoplasm, and we have ribosomes. Those are the 4. Keep them in your brain. Make sure that you understand if we have those 4 things, we have very good evidence that we're dealing with a cell. Now we have 2 types of cells. 1, the prokaryotic cell. It doesn't have a nucleus. It does not have membrane-bound organelles. But it does have those 4 hallmarks. It still has a cell membrane. It still has DNA and RNA. It has cytoplasm, and it has ribosomes, always. Eukaryotic cells also have all those 4, but then a couple of other things as well. They have a nucleus. This is the way I remember it. Euks have a nuke. They also have membrane-bound organelles. They are more complex. Remember, you are eukaryotic. That's how I remember it. And they look kind of like this, although a real cell is a whole lot more squished together, and there's a lot more going on in the inner workings of a cell. Okay, so one of those defining features, one of those hallmarks of the cell is the cell membrane. What is it? Well, it's a bilayer of phospholipids and membrane proteins. We use the term amphipathic to describe the cell membrane, which means that it has both hydrophobic and hydrophilic regions. Think like a frog. It likes water, it lives in water, but it also lives on land. And so this membrane structure is pretty cool. It's constantly moving. It is very, very active. It is going to have 2 layers of phospholipids where these polar, let me grab my PowerPoint, the polar head group is oriented towards the water, which is outside the cell. The nonpolar tails pointed inwards at other nonpolar tails. So this inner part here is my nonpolar tails. Inside the cell is a whole lot of water too. So I need to have 2 layers because otherwise one single layer, these fatty acid tails would be very unhappy. They'd scrunch up. They wouldn't like it at all because they're trying to get away from that water. So if we have 2, well, the 2 hydrophobic tails are able to orient towards each other, whereas the polar heads are able to orient to the water outside the cell and the water inside the cell. Now, we talked about this a little bit last unit, and it comes up again now, that the bends in the fatty acids, aka that double bond that generates a kink, is going to aid in fluidity of the cell. This becomes very important for things like bacteria. Now, like I said, these phospholipids, they're moving constantly, and they can move in a lot of different ways. They can spin. We call this a rotational change. They can move laterally, meaning they can swap places with other phospholipids. And very, very, very rarely, they can do a flip-flop. They can completely rotate around. That's very rare. But it does happen. So this is a very active, it is very lively in the sense that things are constantly moving. Okay, so we said that the membrane fluidity can be regulated in part by having more unsaturated fatty acids. Remember, unsaturated are the ones that are going to have that bend because of a double bond that generates a kink. And this is all stuff from last unit, but it has a double bond that generates a kink, making it not stackable, so it is going to be more fluid. And so, looking at these images here, the one that is going to have more unsaturated fatty acids is going to be more fluid because it's less stackable. Whereas something on the right side, where we have more saturated fatty acids, it'll be less stackable, meaning it's going to be a little bit more solid. So in cold months, a bacteria potentially would prioritize having unsaturated fatty acids in its membrane so that it stays fluid. It doesn't become completely rigid. This is a really awesome mechanism that bacteria use to regulate their cell membranes and their fluidity. Now, we have something else called cholesterol. Cholesterol is a lipid. It is a steroid. How do you know it's a steroid? Well, this suffix is a really good indication. Sterol typically indicates that we are dealing with a steroid. And remember, steroids are these 4-membered rings. And in this case, cholesterol inserts itself into the cell membrane and makes the cell membrane less rigid, aka more flexible. Now, if you have high cholesterol, this is usually not a good thing. And you might go to the doctor, and the doctor will typically recommend eating more fruits and vegetables. And the reason why is because there is no cholesterol in a plant cell. Plants do not use cholesterol. So, switching to a more plant-based diet eliminates dietary cholesterol from the equation. Whether or not that makes a huge difference, it's relatively clear that it does help, but how much is still up for debate. Okay, now, we call the cell membrane selectively permeable. Selective means like choosy. Not everything gets in. So some things get in, some things don't. Permeable means to allow in. So the things that get in easily are nonpolar molecules. Things that do not are going to be polar molecules. And there's a slight mistake on this slide. I want you to be very careful. It's not just need energy, it could be a protein. Ooh, this is terrible handwriting. Protein. Okay. Could be either one. So don't immediately eliminate an answer simply because it says it doesn't require energy, because we have something called an aquaporin that is for polar molecules, for water, and that does not always need energy, it's just a protein. And so looking at this picture, this is one of my favorite pictures in your notes this unit. It's really, really nice. It shows you how things get in and out of the cell, whether or not they're going to get in or they're going to get bounced back, and how they're able to travel in. So that might have just given you a hint that the membrane is not just phospholipids. It sort of serves as the base, but it is not just phospholipids. In fact, the membrane is about 50% protein by weight. 50%. That's crazy. Not just one protein, lots and lots of proteins, but 50% protein by weight. So this is what we call the fluid mosaic model. So remember, we call the cell membrane fluid. We work really hard to make sure that it is fluid. For us, we use cholesterol, right? But for bacteria, they might use more unsaturated fatty acids. But that's the fluid part. And then the mosaic part is that it's not just a bunch of phospholipids. In fact, remember, it's only 50% phospholipid by weight. The other 50% is going to be primarily proteins. So, yes, there are other things there. We see carbohydrates, and we see maybe a glycolipid here, but this is a very small proportion compared to proteins. So proteins are the vast majority of what makes up the mosaic in the basically the base of phospholipids. Okay, these membrane proteins, they have lots of different functions. We're going to talk a lot about transporting proteins in a moment, which are going to help regulate what enters the cell. But there's a bunch, and you're responsible for all of them for this unit and for the rest of the year, because we do build upon these proteins throughout the whole year. So they could be potentially electron and proton transporters. These are going to be really important when we get into cellular energetics in Unit 3. We're going to talk a lot about channels and transporters today. And then we also have receptors and enzymes that become a really important part of cell signaling, so Unit 4 and beyond. So there are a couple of terms, and remember, this is a very vocab-heavy unit, so please be using those Quizlets that I've made for you to ensure that these words are locked in before that exam. So an integral protein, those are the ones that are embedded in the membrane. Peripheral proteins are ones that can attach to the membrane, but they also, go off on their own sometimes. So they can be really important for signaling, and they can have a variety of roles, but these are general terms. We're going to look at a little bit more specific, and then we're going to even look at very specific examples of proteins. So the first proteins that I want you to remember are glycoproteins. Glycoproteins, these are not transporting proteins. They are going to be doing a very different role. They are very important for our immune system to recognize them. So remember, this acts like the flag. It goes, hey, don't eat me, to all of the immune system cells. So something like a T cell that's coming around, or the phagocytes that are coming around, and they're trying to find foreign invaders, things like bacteria. They're looking all the time for that. If you don't have the proper glycoprotein profile, aka the correct glycoproteins, then your cells are going to be subject to attack by those immune system cells. So these glycoproteins act like a flag saying, I'm you, don't kill me, please. They are very important. And you have a very different glycoprotein profile compared to anybody else on the planet, unless you have an identical twin. So that means that if you ever have to get an organ transplant, typically, in fact, always, they are going to also have you be on an immune suppressant for the rest of your life. Because if you're not, then your immune cells will attack that donor transplant. Not ideal, but at least you get an organ. But it does mean that for the rest of your life, you're basically immunocompromised. Okay. Now let's talk about a couple of transporting proteins. Channel proteins are acting like a pore. So they span the membrane and they allow molecules in to move with the concentration gradient. So remember, one of the major differences between a channel and a transporter is that channels are typically things that go with the concentration gradient. There is no energy required. This is for facilitated diffusion. Carrier proteins are very similar, except for they are not going to be constantly open. Instead, remember, I put my hands like this in class. So they have like an open conformation. Solute comes in and delivers it to the other side. So it's going to snap back then to that V shape and let it into the other side. That is what a carrier protein is doing. Then we talked about cell recognition proteins. This is like the glycoproteins that I just gave you an introduction to. These are often glycoproteins or glycolipids, typically glycoproteins. Now they are part of something called the MHC, or the major histocompatibility complex. And they're going to be very important for our immune cells to recognize self from non-self, aka pathogens. Then we have receptor proteins that are going to be very important, again, in that cell signaling unit, unit 4. So be prepared to see them again. This is where they're, instead of delivering something into the cell, they are going to be delivering information into the cell. So some signal is going to be received by the cell. There will be a receptor protein, and then it will bind with some sort of substrate, and that is going to deliver information. It could be information to do apoptosis. It could be information to make a protein because there is a surplus of some sort of sugar. It could be any variety of signal. All it's doing is the signal is telling the cell to do something. Enzymatic proteins in the membrane are still just enzymes, so nothing special here, but know that enzymes can be bound to the membrane rather than in the cytoplasm. And then junction proteins, this is basically when two cells come up to each other and do a little kiss. And the little kiss is going to communicate some sort of information. It's kind of like a receptor to another receptor from two different cells. So maybe if this is a cell that shouldn't be this close to another cell, that could tell the other cell that it's potentially cancerous. And so both cells will end up doing apoptosis to hopefully prevent cancer from proliferating inside the system. So can be really, really helpful. It doesn't always have to be cancer, but that is just one example that it might occur in. Okay, so this is the last piece that we're going to talk about today. We talked about something called the extracellular matrix. Remember that this prefix, extra, means outside. So outside the cell matrix. This is a meshwork of proteins and polysaccharides outside of the cell membrane. And it's going to be really helpful for a handful of things. One, communication between cells. It's going to connect the cytoskeleton through transmembrane proteins to the extracellular matrix, and it will hold tissues together. So cells actually don't want to butt up to other cells. That is not the ideal state. Instead, they want to bind and anchor on to the extracellular matrix. And all of these different types of cells, or all of the cells of the same type really, are going to link together to this extracellular matrix to basically create a web, rather than all of the cells just touching next to each other. The proteins that are going to anchor the cytoskeleton inside the cell to the extracellular matrix outside the cell is a protein called integrin. Integrin is that anchor protein that links the cytoskeleton to the extracellular matrix. Now, a protein you've probably heard of in the extracellular matrix is collagen. Collagen is a protein that gives this extracellular matrix strength. So as collagen begins to break down, that means that this webbing between cells is getting looser. And so that's when we get sagging, aging, people looking old and wrinkly. Things don't snap back the way they used to because the collagen is degraded. So practical application, right? And so there's a lot of people at some point, they are going to try to exploit your fear of aging and looking old. And they're going to try to give you collagen supplements that you take, or you put on your skin. And I'm going to tell you now, they don't work. Okay, there's no evidence for that. What you can do to prevent aging, not much. You're going to age, but you can wear sunscreen. So sunscreen is a really great protective factor against collagen degradation. So it's very important to wear sunscreen. And that's what I'll leave you with in this review. Good luck.